Alright, welcome back. We just talked about carbohydrates, now we're going to have a quick look at the lipids. Now, the larger group, or the big subset if you like, is lipids and these macromolecules, because many people interchange lipids and fats, but that's not quite accurate because fats are a type of lipid. When we talk about lipids, we are including both the fats and the steroids, and they are structurally different, and therefore because they're structurally different they also have different properties. So behind me I put the two groups, the fats and the steroids, which make up the lipid group. Now in fats and steroids there's no true monomeric part. To get into the body they have to be broken down to their smallest point. The cholesterol etc. can come through, but the triglycerides etc. get broken down into their component part, which is the glycerol head and then the fatty acid tails. The same would be true for the phosphate head. So these are fatty acid tails. Crazy S there, take that off. So, we don't have any true monomers of the fats or the steroids, unlike the carbohydrates, the proteins and the nucleic acids. It's one difference between them. Like the carbohydrates we just looked at, they're made up of carbon, hydrogen and oxygen. But this time they don't have a clear ratio. Carbohydrates had 1:1. In the fats and the steroids, the carbon and hydrogen is dominant. There's many of these, and there's only a very few oxygens. Now this is important if you remember your chemistry, because in the last section, the carbohydrates, if you remember glucose, fructose had C6H12O6. Well, that's 6 oxygens for 6 carbons. And if you remember, covalent bonding, the polarity, or the ones that had that slight charge, were dependent on the presence of oxygen pulling the electrons away from the hydrogen. So carbohydrates carry that kind of polar covalent bond because they have a lot of oxygen molecules, and the oxygen molecules are doing the same to the hydrogen and also to the carbon molecule. Compare that with the fats. There's very few oxygen molecules in a large fat, a big long triglyceride with a long fatty acid tail. The oxygen really only exists in the join between the fatty acid head and the glycerol itself. So there could be as little as 3 oxygen molecules in the whole thing. And that means that up here, so there's like an oxygen, the oxygens you find are here, here and here. And we've got some, I haven't written all the structures in the top, you can look that up if you're interested. And I've just drawn the carbon chains. There's hydrogens attached onto here all the way down, so hydrogen there, hydrogen there, hydrogen there, hydrogen there. Oops, attach that onto the bond, so that's all the way down. So this is all carbon-hydrogen. These are all non-polar. And up here there's a little bit of charge created by that. The phosphate head is a P with oxygens around it, and they're all negative. So the two heads of these different types of triglyceride and phospholipid have a little bit of charge on them, but these do not. These are hydrophobic. So fats in general do not dissolve in water because they don't have enough charge on the molecule as a whole to interact. So they act in a hydrophobic manner. So lipids are hydrophobic because they carry so little oxygen and therefore do not develop polar covalent bonds. So even though they have the same ingredients, the lack of the oxygen leads to non-polar. So that leads to non-polar covalent bonds. So no overall charge. And that equals hydrophobic, therefore does not work with water. Alright, so again, their specialist bond occurs, because they're not monomers, occurs between the head of either the glycerol, the phosphate head here, the triglyceride or phospholipid, depending which one we're looking at. Phospholipids, by the way, are found in the cell membranes and the membranes of our organelles in our cells, as we'll find out when we look at the next section. Glycerol heads and triglycerides is what we consume as fat in our diets. But they're both joined in the same way by an ester linkage. Remember, in carbohydrates the monomers are joined together by glycosidic linkages. In this case it's an ester linkage which joins the long carbon-hydrogen tails onto the head, either the phosphate or the glycerol. And that's what's broken when they're broken into their component parts ready for absorption in the gut. So when you break them down, if it's a triglyceride, you get a glycerol head and three fatty acid tails, because it's a triglyceride. And then if it's a phosphate, then it would be phosphate head and two fatty acid tails. On the steroid side, the base steroid is cholesterol, and that's what we consume in our diets. But our livers also have a pathway for making it if we don't take any through our diet. So we can both make it and we can consume it in our diet. And the statins that we take to lower our cholesterol levels are actually targeted at what we make in the liver. And then of course you're advised to keep your cholesterol under control in your diet as well. But again, research is showing that a lot of our dietary cholesterol is not necessarily closely related to our actual cholesterol levels in our blood. So cholesterol is then used to make all of the other steroids. So the sex steroids like estrogen and progesterone, testosterone, aldosterone, which we'll look at in endocrine, and the corticosteroids as well. So all of them are made from cholesterol. Now, whilst this is all long fatty acid chains in the head, cholesterol is a four-ring structure, and these are also four-ring structures because they're based off cholesterol. So structurally very different. So cholesterol is used to make the other steroids, which are signal messengers in the body, but also you find it in cell membranes as well because it helps to hold the phospholipids more tightly together. So phospholipids are there to create the bulk of the cell membrane and the organelle membranes, so to create spaces, specialist areas or rooms if you like in the cell. And triglycerides are there to produce energy. The fatty acid tails can be put into the respiration pathway the same way as glucose can, and you can derive ATP from that. Storage of fat, particularly subcutaneously under our skin, allows us to stay warm as well. And fat also around our organs helps to protect them from buffeting, but too much fat around our organs interferes with their function. And a common way of developing type 2 diabetes is essentially creating a fatty liver, which then spreads over and creates a fatty pancreas and affects the insulin output. So lipids break down into fats and steroids. They're all hydrophobic because they create mostly non-polar covalent bonds between carbon and hydrogen. There's a few oxygens but not many, and that therefore doesn't help them interact particularly well with charged particles. They're not monomeric and they don't have a clear ratio between the carbon hydrogens and oxygens. The two parts, fats, break into the triglycerides and phospholipids. You could also put the waxes in there as well if you're interested in that. And triglycerides have three fatty acid tails, phospholipids have two, with a phosphate group coming off on the third side instead of another fatty acid tail. They're joined by ester linkage, and the level of saturation is one thing I haven't talked about. So the level of saturation determines if a fat is liquid or solid. So you know that olive oil, for example, is liquid at room temperature, but butter is solid. And this is all down to the fatty acid tails here. If they're all fully saturated, and to be saturated it means that you have one bond between each of the elements involved. So carbon can create four bonds, and they're all used. So there's oxygen there, hydrogen, hydrogen, carbon to the next one. So they're all tightly packed in. So as you tightly pack something, the more solid it becomes. However, if, for example, that one's a double bond, any double bond in the molecule makes it bend, and then that then joins onto hydrogens there. and that causes a bend in the carbon. Actually, I'll put it there, put that there, and take that off. And if you have lots of these, you end up with a molecule with a tail kind of like that, and you can't pack two of them very close together, whereas you can get two with straight lines very close together. So that's why at room temperature animal fats such as butter, lard, etc. pack in tightly, because they're all saturated, and unsaturated or mono unsaturated fats have all these kinks in their fatty acid tails which push them further apart, so just make them a liquid at room temperature. That's pretty much everything I want to say about lipids, so we'll stop and then move on to protein.